Kita nanti sudah bukan yang kita punya dua.
දැම්මා ගුපට දැම්මා.
Right. Good evening, people. Good evening. Can you hear me clearly? I know other. Do you hear me clearly? Am I audible? Lamai? I know other. Yes, sir. Abdul, there's a small technical issue. There's a small lagging here. Lagging या क्या नहीं? लोको lagging या क्या? मुझे तो ना सकते कहते. बोर्डे पाँच करने में ने लग गया ना? नहीं गत लग गया ना? बोर्डे फ्री से ना? कौन लग गया ना? Sorry about that. Small technical issue. There's a lagging. I think it's okay now. So we're going to discuss paper number 2 today. Paper number 2. Did you answer paper number 2? Did you answer paper 2? Lamai, answer kara the paper 2 week. Right. Get the find your answered papers. You have to upload that. Huh? You upload it. We'll mark it for you. Right. Upload the paper. Right. Last time, people, we learned we discussed the protein and nucleic acid part. So let's go with the questions. Let's go with the questions. Question number 1. Which of the following contain elements? Carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Which of the following contain? Which of the following contain elements? Okay. Right. We'll go to the board. Right, people. Which of the following contain elements? Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Carbon, hydrogen, oxygen, nitrogen, and phosphorus. Phosphorus and sulfur neither. So phospholipids, carbon, hydrogen, oxygen, nitrogen, but phosphorus, but no sulfur. Phospholipids don't contain sulfur. So therefore, we can't take the first one. We can't take the first one. Second question, people. DNA, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Again, sulfur not good, ne? Sulfur not good. But it says carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. Methionine is carbon, hydrogen, oxygen, nitrogen, and sulfur, ne? No phosphorus. No phosphorus. No phosphorus. Then chitin, carbon, chitin, carbon, hydrogen, oxygen, nitrogen, neither. Hemoglobin, carbon, hydrogen, oxygen, nitrogen, sulfur present. Phosphorus absent. People, there is a weakness in that question. I find that question has a weakness. So please, yeah. Okay, so phospholipids, people, if you take phospholipids, phospholipids contain nitrogen. Choline part is there. So phospholipids, if you take the components of phospholipids, carbon, hydrogen, oxygen, nitrogen, choline part is there, and phosphorus present. We can't take sulfur, as I remember. Yeah, so we can't take sulfur. So we have a small trouble with the first question. Carbon, nitrogen. If you check the diagram, people, nitrogen present, carbon, oxygen, phosphorus. So you can't take even choline. You can't take even choline. So phospholipids, no. DNA, carbon, hydrogen, oxygen. We can take carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur also there. Sulfur gonna ban. So DNA also we can't take sulfur. Methionine, sulfur containing amino acid. Methionine. Chitin also wrong. Hemoglobin also wrong. No answer for that question, people. Right? Put a question mark. Right? Put a question mark. So no proper answer for that. Right? And next one, people. Second question. Second question. Which of the following is the least available order and organization show all characteristics of life? Which of the following is the least level? Which of the following is the least level of order and organization? Order and organization show all the characteristics of life. What do you take, people? Organelle, cell, macromolecule, organ, tissue. I think we can take cell neither. And order and organization shown all of the characteristics which are the following is the least level of order and organization. So we can take least level of order and organization. Show all characteristics of life. So we'll take people second one as the answer for that. Cell is a better answer. Ne? Question number 3. Question number 3. Which of the following property of water is favorable to aquatic organisms in polar regions? Aquatic organisms in polar regions. Did I give you the marking scheme? I forgot to give you the marking scheme. Wait. I'll prepare the marking scheme to the chat. Oh, just give me 1 minute. I'll add the marking scheme to the chat. Right. Check that part, people. That's the paper and the marking scheme. That is the paper and the marking scheme. All right. So, question number 3. Which of the following property of water is favorable to aquatic organisms in polar regions? Property of water is favorable to aquatic organisms in polar regions. Versatility as a solvent, expansion upon freezing, biological medium, high heat of vaporization, cohesive behavior. Shall we take the expansion upon freezing? I think second is a better answer. Eh? Second is a better answer. Second is a better answer. Question number 4. Question number 4. Which of the following statement regarding given structure of the molecule is above is correct? I think people, this structure is missing. I ask you to draw this. A sugar molecule. I ask you to draw a sugar molecule like this. Right. Let's take that as the structure CH2OH. CH2OH. OH. This one. Eh? Right. This one. Right. Which of the following statement regarding the given sugar molecule of above is correct? It's a non-reducing sugar. Non-reducing sugar. So give sucrose with glycoside bond. Okay, we'll see. Fourth one, Larry. Non-reducing sugar. Non-reducing sugar. No, this is a reducing sugar. And it is a constituent of nucleotide. The ribose is no wrong. So non-reducing constituent of nucleotide wrong. And then it's involved in formation of polysaccharide. Involves in formation of polysaccharide. Not involving wrong. Wrong statement. Give sucrose with glucose by joining with glycoside bond. That is correct. And then people does not give positive reaction with Benedict reagent. That is also wrong. So we got the answer here. Fourth one is the answer. Question number 5. Which of the following combination of polymer and its function is correct? Combination of polymer and the function is correct. Right? Polymer and the, which of the following Which of the following polymer and the combination is correct? Lactose storage. Lactose is not a polymer either. It's a disaccharide, so you can't take that. They are asking function is correct. Lactose storage, but it's not a polymer. So you can't take the first one as the answer. Inulin transport. Inulin sucrose is transported. So that is wrong. Inulin storage. And then fat respiratory substrate. Fat is a respiratory substrate, but it's not a polymer. You can't take that also as correct then. Wrong statement. There is a lagging in my side video. I think I have to find that. Inulin blood glucose level, homeostasis. Sorry, insulin. Insulin blood glucose level homeostasis. That is important and that is correct. And then glucagon catalyst. That is wrong. Glucagon catalyst wrong. So we'll take fourth one as the answer. Fourth as the answer. People, fourth one as the answer. Question number 6. Which of the following joint incorrect regarding substance and function? Which of the following joint is incorrect regarding substrate and function? Joint can combination. Cut the word joint and take it as combination. Is there any disturbance to the audio, people? The video is lagging. Video is lagging. But is any disturbance for the audio? Okay, audio okay. Which of the following combination incorrect regarding substance and function? Sucrose storage. Sucrose storage. Yeah, in some plants, sucrose is a storage material, sugarcane and all. And chitin structural support. Chitin correct. And collagen mechanical support. Collagen correct. Maltose transport wrong. Suberin limiting permeability. Suberin limiting permeability correct. It's present in some plant parts. So we'll take people maltose transport is wrong. Fourth one for the sixth question. Moving to question number 7. Which one of the following joint is, which of the following joint can combination? Which of the following combination regarding substance found in living organism is incorrect? We'll take combination once again. Combination. Which of the following combination regarding substance found in living organism function is incorrect? Sucrose storage, pectin structural component. Sucrose storage correct. Pectin structural correct. Inulin mechanical support. Inulin mechanical support wrong. The inulin is storage. Glucose transport wrong. Suberin limiting permeability correct. So we have two answers there. Inulin mechanical support wrong. And then glucose transport is also glucose transport. How about people taking glucose transport in animals' body, your body glucose transport? Your blood glucose circulate. So glucose is circulated. So I think the best answer then, third one. Inulin mechanical support is wrong. So we'll correct the scheme also. Seventh one, people. Seventh one, third better. Seventh one, third better. Seventh one, third is better. Right? You mark third. You mark third. Dylan. Third is markery. That's good. And then people, question number 8. Question number 8. I'll move to next page. Question number 8. Question number 8. The question based on following bonds. Ionic bonds, hydrogen bonds, phosphodiester bond, peptide bond, glycosidic bond. Which of the above bond found in both proteins and nucleic acid? Proteins and nucleic acid. Ionic bonds you can't take, ne? Proteins and nucleic acid. And then hydrogen bonds, proteins and nucleic acid, you can take that, correct. Phosphodiester bonds, proteins you can't take, nucleic acid available. Peptide bond, protein present, nucleic acid absent. Glycosidic bond, glycosidic bond, glycosidic bond you can't take, ne, in polysaccharide. So we'll take answer as only B. Fifth one is the answer. Question number 9. Question number 9. Which of the following is not a globular protein structure? Not a globular protein structure. Hemoglobin globular, neither. Hemoglobin globular. Globulin globular. Keratin is not. Myoglobin ball-like. Amylase is an enzyme and globular. So we have the answer, third one. For question number 9, third one. Question number 10. During denaturation of proteins. During denaturation of proteins. What is protein denaturation? What is protein denaturation? The natural conformation of the protein lost, that we call as protein denaturation. During denaturation of proteins, loss of their secondary and tertiary structure. Secondary tertiary structure denaturation can be lost, possible. Loss of their primary structure, Varadi, they maintain the primary structure. Loss of their peptide bonds, wrong. Peptide bonds are primary structure and get solubility in water, wrong. Lost their disulfide bridges. Denaturation, during denaturation, lost their disulfide bridges. Possible hai bhai. Disulfide bridges possible, but does not happen most of the cases, right? Lost secondary and tertiary structures, lost their disulfide bridges. This is also correct, ne? Disulfide bonds can break during denaturation of proteins. So I think I have two answers, right? So during denaturation, right, denaturation of proteins, we'll take people disulfide bonds, secondary tertiary structures loss means that's it. First and 5. I'll take two answers for that. I'll take two answers for that. The market scheme I have given 10th question 1 kilo. 1 and 5 also possible. But I think in the resource book, what it says, loss of specific three-dimensional shape as a result of alternation of weak chemical interactions. But that specific three-dimensional shape means the tertiary level structure is lost by breaking of disulfide bonds, ionic bonds, other interactions they can break, right? Sulfur bonds also can break. I think we can agree with those two answers, 1 and 5. Question number 11, people, the base adenine of DNA of an organism forms 13.3. Adenine 13.3, look. Adenine 13.3, right? Adenine 13.3, adenine is equal to thiamine. Both are 26.6. Guanine cytosine, guanine plus cytosine, remaining amount, the base of adenine of DNA is of an organism 13.3 in its composition. Percentage of purines. They ask for purines. Purines always, people, 50%. You don't have to do any calculation. I didn't read the question. Purines always 50%. No need to go for this calculation. Question number 12. Question number 12. Which one of the following characteristics of water directly responsible for the absorption of water and minerals from soil solution? Following characteristics of water: polarity, chemical property, high adhesive and cohesive force, liquid at room temperature, high specific heat capacity. What do you take, people? Third one is better, ne? Adhesive, cohesive features you can take. Question number 13. Question number 13. Which of the following can be seen in both animals and fungi? Which of the following can be seen in both animals and fungi? What is your answer? Lamai, who's going to answer? Santosh says chitin kela. And then some people, Dylan says second kela kena. Which of the following can be seen in both animals and fungi? That is correct, ne? Santosh, Dylan, you all are correct. Chitin you can take as the answer. Question number 14 and 15 are based on these combinations. A, B, D, you have to select one. A, C, D, second. A, B, third. C, D, fourth. And then any other combination. So we'll go for question number 14. Question number 14. Which of the following is are not structural polymers? Which of the following is are not structural polymers? Nucleic acid is not structural. Ne? Starch is not structural. Answers are not kilahane. Not structural polymers. Right? Which of the following are not structural polymers? Polymers. Structural. Polymers. Nucleic acid. Nucleic acid is, how do you take nucleic acid? Right? Store genetic information. But then it forms a DNA and chromosomes. Ne? Starch is storage. Ne? Starch is storage. Pectin is structural. And then inulin is storage. Chitin is structural. Not structural ke lahanne. So pectin you can't take. Chitin you cannot take. And then starch is correct. Inulin is correct. Nucleic acid is the question. Nucleic acid is the question. Following are not structural polymers. Structural. Now, when we take nucleic acid, people, it's forming DNA molecule, chromosome structure, building up of chromosomes, chromatin. But it's involving in storage of genetic information too. Right? So, structural. Functions store and transmit genetic information, store genetic information for protein synthesis, structure of the DNA molecule. We can't take it as like building up of particular structural component, right? So building up of particular structural component. So we'll take it as people, it's store genetic information, right? So I'll take first one also as correct. So answer is A, B and D kilogam. Answer is, then I'm changing the marking scheme one, right? So we'll take the answer once again. ABD kilogam. ABD. Right? ABD. Then answer goes to ABD fifth one. Sorry, ABD first one. First one is the answer. Right? And then people, which of the following statements regarding cellulose is are correct? Statements regarding cellulose is are correct. It is found in some prokaryotic cells. Found in prokaryotic cells. Statement regarding cellulose. Prokaryotic cells are wrong. And then it can be hydrolyzed by amylase. Cellulose wrong. Present in cell wall of chlorophytes. That is correct. And then numerous glucose molecules are linked by glycosidic, correct. Storage polysaccharides in some. Polysaccharide in some. No. So CD must be the answer. CD must be the answer. 15th question. 15th question. Answer is CD. Right, moving people to the structured questions. Moving to the structured questions. Moving to structured part. Right. Moving to structural part. Structured questions. Right, first one. State elements present in minute amounts. Elements in minute amounts. What we said, people? Kokro mage no call kokro mage beef kufe ne. No call ne. No call ko crow mage BIF kufe ne. Beef kufe. No call crow mage beef kufe. What are trace elements? Elements present in the body which are less than 0.01% of dry mass. Okay. So trace elements. State the intermolecular interactions between water molecules. Hydrogen bonds present. Intermolecular interactions between water molecules. Hydrogen bonds. Ne? And then next one, people, how water film forms in an aquatic system. How water film is formed in an aquatic system. Right? The upper surface water molecules are attracted by the lower surface water molecules due to cohesion between water molecules. So we'll take that as the answer for that. Question part 3A. Name carbohydrate groups. Name carbohydrate group. Name carbohydrate group. Name carbohydrate group. Where all sugars are reducing and water soluble and crystalline. Where carbohydrate group where all sugars are reducing, that is then definitely monosaccharides. Monosaccharide must be the answer. And then on what basis the aldose and ketones are differ from each other? On what basis aldoses and ketones are differ? Carbonyl carbon, right? Carbonyl carbon. The aldo and the keto carbonyl carbon. I think you better write it like that. Aldo and keto carbonyl carbons. And then next one, people, what are sugars available? What are sugars available in ring form in an aqueous medium? Glucose and fructose. Glucose and fructose, we'll take that as the answer. State why sucrose is non-reducing while maltose and lactose are reducing. In sucrose, both carbonyl carbons, they are involving in engaging in formation of the peptide bonds, whereas in the maltose and lactose, there is one free reducing end present. I think you better say that the both carbonyl carbons form glucose and fructose. In sucrose, both carbonyl carbons form glucose and fructose. Let's say that both carbonyl carbons are involving in forming glycosidic bond. Make a small correction. Both carbonyl carbons are involving in the formation of the glycosidic bond, right? But in maltose and lactose, free carbonyl group is available. So therefore, it can undergo reducing. State sugars present in polymers other than carbohydrates. Sugars present in polymers other than carbohydrates. You can take ribose and deoxyribose. Ribose present in RNA, deoxyribose in DNA. State the difference between nucleoside and nucleotide. Nucleoside is sugar, nitrogenous base, and the sugar we say nucleoside. Nucleotide, nitrogenous base, sugar, and phosphate part. Next one, we will give two examples for nucleoside. Adenosine, guanosine, you can take as examples. Describe the way of forming sugar phosphate backbone between two nucleotides. Describe the way of forming. What happened now? The third carbon OH group react with OH of the phosphate in forming the phosphodiester bond. In forming sugar phosphate backbone, two nucleotides from phosphodiester bonds by condensing between the OH of the phosphate and of one nucleotide and OH attached to the third carbon atom of the pentose sugar of the other. That is what you have to write. And next one, people. Describe the way of forming sugar phosphate backbone. Okay, done. Why polynucleotide strands of DNA are said to be antiparallel? Why polynucleotides strands of the DNA are said to be antiparallel? The two sugar phosphate backbones run in opposite direction. That is what you write. What makes the two nucleotide strands of the DNA complementary to each other? Always purine base pair with specific pyrimidine base, right? That you can take as the answer. So, people, essay question I have asked this time, describe the structure of the DNA molecule according to Watson and Crick model. DNA structure according to Watson and Crick model. Check that part, people, the marking scheme. Check that marking scheme, right? So, describe the structure of DNA according to Watson and Crick. So, you have to see that the DNA consists of two polynucleotide chains. DNA has two polynucleotide chains, which forms a double helical structure. These chains run opposite direction. So, they are antiparallel. Nucleotides are arranged linearly in polynucleotide chains. Nucleotide consists of three parts: nitrogenous base, deoxyribose sugar, and phosphate part. Two types of nitrogen bases are there, purines and pyrimidines. And purines are adenine and guanine, pyrimidines are cytosine and thymine. Adjacent nucleotides are linked through phosphodiester bonds, forming sugar phosphate backbone, linked through complementary bases. Adenine binds with thymine and by 2 hydrogen bonds. Guanine binds with cytosine by 3 hydrogen bonds. And then you have to draw the diagram. So this is the expected diagram. You don't have to draw the diagram like this, even if you draw the diagram, people, on this way, right? They have given marks. Look at this, give marks for this diagram also. So you can say adenine, thymine like that. 2 hydrogen bonds, guanine, cytosine, 3 hydrogen bonds, 3' end, 3' end, 5' end, 3' end, 5' end. Sugar phosphate backbones you can label. You can draw the diagram simply. Right? So that's the end of our today's discussion, right? So thank you very much. I'll give you the next paper, third paper on Saturday, right? Monday, as usual, the our extra class, right? So then, thank you very much for your participation, and we'll be meeting once again soon. Salute for all of you who joined with the class today. Okay then, let's see you. Bye.